Hi, I'm Rob.
I'm a gastroenterologist and I'm passionate about gut health.
Hi, I'm Kaz, and as a new mum, my family's health is important to me. I've heard the secret to well-being lies in your gut.
But these days, it's hard to know what to trust online.
Luckily for me, my friend Rob is a leading Australian professor. And for 20 years, I've been picking his brain.
Is leaky gut a real thing? Is the microbiome really the key to good health? Don't sausages cause cancer?
It got me thinking, if Kaz wants to know more about this stuff, others might too.
So together,
We're going to share the latest cutting-edge gut facts with you.
We have the real ability to revolutionise medicine.
Our experts will be busting myths,
No, nonsense again.
and we'll be taking the tests
What have I got myself into?
to tell you
the gut truth. Welcome. How good is it to be here in the basement?
it's exciting.
Really exciting. And we've got Sammy Forster with us, someone I know really well. He is a world legend in terms of microbial science. Sammy has cultured the unculturable organisms and has a huge microbiome culture collection. And he's here today to talk to us about the gut microbiome.
Yeah, great to be here. Thanks, Rob.
Welcome, Sammy.
I'm interested to know what made you want to work with microorganisms.
So actually, I wasn't a microbiologist to start off with. I started off as an immunologist, but I realised we have these microbes that we all carry with us, and we needed to understand them. There's an opportunity, if we understand them, to make new medicines. And back then, we couldn't even grow these things in the lab. And so it's
Really?
about being able to grow them, being able to understand them, and then being able to give them to people as new medicines.
Sammy, so what is your microbiome and why does it matter?
So we all have these microbes that we carry with us every day. Viruses, bacteria, archaea, different types of microbes. And they're critical for our health in a whole range of ways. They exist in our lungs, on our skin, female reproductive tract, the gut. Obviously, we're focused on the gut today because that's where you decided to hold your podcast. But
Yeah.
fundamentally, though, the gut is a really important site. There's a huge number of bacteria, roughly the same number of bacteria as human cells, a huge amount of function, so 100 times more functional capacity in the microbes that we carry than our own cells.
Wow.
There's a huge functional capacity there that provides health benefits, can be detrimental.
Yeah. So you've been a big part of this journey of discovery of the gut microbiome in the last 10 years.
I've certainly been part of the journey. I mean, there's
Yeah.
a lot of work going on all around the world because
Yeah.
we see this potential,
Yeah.
but the fundamental science is still lacking. And it's crazy. We carry these species around with us. Every single one of us has these species, and we just don't know about them. We're still really trying to understand what they do. There's different phyla in there. That's the difference between a vertebrate, something like us, and a mollusk. There's huge diversity, and we just don't understand it. We have to understand in the same way as you would if you were studying any environment. You can't just look at an environment and suddenly know what it does. You need to understand the individual parts, whether that's a forest, a coral reef. You have to understand what's there. And that's the first bit of what we're doing.
Sam, you've described this journey that you've been on since 2013, and a lot of it relates to the technology and the ability to classify and identify organisms right down to a much more refined level, a deep dive as it were. Can you tell us about that?
Yeah, I mean, so we've worked down, the technology's got better, we've got better understanding of the individual species and even the isolates that are present. So we can sequence the microbiome and understand all the species in there through the genetic code.
And so what are some of the species that live in our guts? Can you tell me a little bit about these microorganisms?
We have a huge variety of organisms that live in there. We focus on the bacteria. That's been an area that we've done a lot of work on, obviously. But there's other things. There's archaea, there's phage, so bacterial viruses, there's fungi, and then there's other eukaryotes, so things like parasites. Less common in the Western world, but certainly historically things that have been there.
But we mostly think of bacteria, don't we?
We do, and the bacteria are a major part of the community.
We've spent a century trying to kill them, really, with
Yes.
hygiene and antibiotics, which has vastly improved our quality of life, our longevity, but now I think the tides have turned.
Well, we focus on the pathogens.
Yeah.
Everybody is aware that you go out for a meal, you wake up the next day, you don't feel great,
Yeah.
you blame the food you ate,
Yeah.
the pathogen that was on it. No one does the opposite. No one ever thinks, I'm feeling great today, it was the bacteria on the food or the bacteria I encountered in the environment. But actually, the majority of species we encounter are health associated or just don't have any effect.
Yeah.
And there's lots of different communities that exist through our gut. And so while we're typically thinking of it in terms of the whole gut, we know within the gut the stomach is going to be different to the colon, for example, the nutrients that are available, the environment, and therefore the species that can live there. In the same way as some species will live in a forest and some will live in the ocean. Yeah. And so all of these sites have different communities. Some of the species are uniquely specialised to living in one of those spaces, but they're all important, they're all providing function.
So you're saying there's a microbiome that's different throughout the gastrointestinal tract, different in the stomach to the small intestine to the large bowel. What do we mostly measure then?
So we measure fecal samples, usually through sequencing like you have with the gut tests.
Have you ever done a microbiome test, and what do you think of microbiome tests? This year I've been more sick than ever before in my life, and I've seen so much online about the importance of the microbiome to your immune health. So I decided to order an at-home microbiome test kit. Do you think that was a good idea?
I'm really sorry to hear about your year, Kaz. I wouldn't usually recommend it. I mean, there's a lot of gut microbiome testing labs out there. I see a huge variability in terms of quality. The good thing is these days there are some better accredited labs where your results will be more accurate and meaningful. Results will be interesting. It perhaps will give us some insight into your gut health.
Are you filming?
Yep.
Because there's just been a kerfuffle at the door and I think something's arrived.
What is it?
It's my microbiome testing kit. May I please have those scissors that you're playing with? Good parenting.
What's in it?
Well, it's a testing kit. Is this an intelligence test?
Oh, she's failing.
Oh, okay, there's an arrow. To unlock health from within, follow these 4 simple steps. Read instructions in full, collect and label samples, select your stool consistency. Oh, there's all these different little bits and pieces. This is quite involved. Feces collection device.
Wow.
Oh wow, I've got to put a band over the toilet
Wow.
so that none of them fall in. Guess they don't trust me to just like close it.
Mama, are we going somewhere?
To the bathroom. Bye.
Have fun.
So I actually haven't done a microbiome test.
Why not?
Because the problem is we really don't understand what different compositions, what the benefit or detrimental effect of a particular composition is. And for any individual, it's not like we can say, you've got this test, you should do X and therefore you'll be better. If you've just taken antibiotics and you have no microbes, we can see that that's bad. If you have a diverse community, that might be a good diverse community for you and a detrimental diverse community for Rob. And so the interpretation of these tests is actually really difficult. That's not to say that they don't potentially have value, but they have to be coupled with the knowledge.
Yeah, right.
How many species are there in the gut?
So for most people, it's a few hundred. There's roughly 1,000 species, give or take, that could be in there. If you're obviously post-antibiotics, that number will be far lower.
So I'm taking antibiotics at the moment, and it's making me wonder what's going on in my system.
It's a good question. What's left after antibiotics?
Depends on the antibiotic.
Right now I'm on penicillin.
Broad spectrum.
In a healthy microbiome, healthy gut microbiome, we should have a dominance of Firmicutes bacteroides, so the 2 health-associated groups. And when you get disruption, and that can be an antibiotic treatment, it can be an immune response, these species become less able to exist. If you're particularly healthy, you have like a diverse lifestyle, diverse diet.
Lifestyle.
Lots of exposure to different things. Most of us work in one place. We go to and from like that one place. We have a few
fairly similar foods week to week.
Yes.
and therefore we get the same bacteria in there. But we know you do more exercise, you do different things, you have different exposures, the microbiome changes. And so it's about the more diversity you have, the more exposures you get, and therefore the difference in species you have. A lot of these microbes, they do spread through the environment.
Yeah.
And if we block some of those transmission routes, we don't pick them up.
Yeah.
And what's the consequence of that?
And that's the health benefits that we lose. The natural process is for these microbes to be moving between us. You want to have the diversity, because if you have one species, it's able to colonize because the gut is a great environment for these species to live. There's nutrients there, there's all sorts of benefits. But you have one species and it takes over. It's just like a locust turning up, like you get this mass growth of one thing.
It becomes a monoculture.
Precisely.
Yeah.
But fundamentally, the most important thing of all is what do they do? What are their functions?
Yeah.
And so there's groups of bacteria that provide certain functions to us, and it's about understanding those and understanding when those functions are beneficial.
I think that's a really important point, that it is function over just composition. I think that's a really, really important take home.
Yeah.
So how important is our microbiome to our overall health and function?
The fact we have a microbiome is remarkable. We all have an immune system, right? If there wasn't an evolutionary pressure for benefit, our immune system would have evolved to wipe these species out. So if you think about what we're doing, we have this community of microbes that's existing in these effectively dangerous positions for us. It's right up against our cells. It's in our gut, for example, where it could break through that barrier, it could cause an infection. Yet our immune system hasn't designed or hasn't evolved a way to remove those bacteria, or other microbes. We allow them to exist there.
Which implies a symbiosis.
Which implies a symbiosis. It also implies that there must be a selective pressure beneficial to have that. And that selective pressure actually has to be stronger than the risk of the pathogens.
Yes.
And we know, I mean, the risk of a pathogen is death. So there's a very strong selective pressure to not have these species there. So the function they're providing must be hugely important.
And what are those functions?
Well, it's an interesting space because originally the thought would have been, well, they're helping us digest food. They're helping us like save those last nutrients that we weren't able to get through our normal digestive processes.
I think that's been the long mantra, hasn't it? Gut digestion.
Yes,
You know, that's what we think
absolutely.
about. And this whole gut health movement and understanding really takes it beyond that.
Yeah. Well, I think the key thing there is we can see in experimental models where you can remove the microbes from the animals, for example, and those animals don't starve to death. So if it was all about nutrients, if it was all about food, why don't they starve to death?
So what else do they do?
And so the thing that happens is their immune system has these remarkable changes. So it doesn't develop properly. They suddenly become susceptible to all of these infections. And so it's about immune modulation. And that takes us back to the original ideas of, well, microbes interact with the immune system.
Yeah.
And it makes sense, right? You have this ability to stimulate the immune system. You have these interactions going on. And if you think about it, one of the biggest limitations of our immune system is we can't see things coming if they're outside of our body. We don't have immune cells that walk around outside our body and pick things up. But the
So it is
microbes are
primary.
there.
Yes.
The microbes can respond to the environment. And if we can sense how they're responding, that gives us an ability to detect incoming threats.
Yeah.
And so it's likely that there's roles there, both at a metabolic level, but also at a direct, like, components of the microbes level.
Yeah.
And that's where the basis of immune microbiome interactions comes from. And what we see, if we think about the diseases that we typically associate with the microbiome, they're actually a lot of immune-related diseases. So we think about inflammatory bowel diseases and things like that, obviously. We think about Clostridium difficile infection, which is the first space that fecal transplant has worked. So a whole range of diseases are associated with the microbiome. It can be local infections, autoimmune diseases, inflammatory diseases. So things that make sense: microbes, immune system, detrimental inflammatory response. But then also more broadly, there's associations with things like gut-brain axis, so autism, Parkinson's, MS have all had associations with the microbiome. A lot of cancers have an immune element as well. It's a failure of the immune system to clear detrimental cells. And that's the local environment. But also, we have this huge area of exposure where the microbes are interacting with the host. We think about our gut as a space for absorbing nutrients, but it's a space for absorbing things.
Yeah.
And if you've got microbes there, you've got the ability for that absorption to happen. And then that can signal to the whole body, because immune cells traffic through the whole body. You can have a local response, but you can have a response anywhere.
The gut microbiota primes the immune system, and that relationship then sets up so many other conditions in health and in disease. Yeah.
Well, as you would know as a clinician, how many diseases have a basis of an inflammatory response or an immune response?
It's emerging that everything comes down to immunity and inflammation. And if the relationship with the gut microbiome is the genesis or the origin of that, then I think that's the story you're telling.
It's not the only part of the picture, but it's a part of a picture we've ignored.
Yeah. Sammy, we talk about this invisible extinction event whereby loss of gut microbes over time is comparable with an environmental disaster similar to what we're seeing in other ecosystems worldwide. What's your take on that?
We've basically spent the last 100 years trying to kill pathogens, trying to wipe out bacteria, whether that be through use of antibiotics, through hygiene and reducing the spread of bacteria.
All of which has increased our life expectancy.
All of which have been has been beneficial, unless you happen to be a microbe, particularly a health-associated microbe. Yes, it's improved our life expectancy, but we've also got all these immune conditions that are now becoming far more dominant.
Yeah.
And the question then arises, well, is it associated with the fact all of these bacteria that were previously modifying our immune system, stimulating responses, are really struggling to spread? Where previously they could have easily moved between people, we've put all these barriers up. We've gone to stop the pathogens, to stop the 1% of species that are pathogens.
Yeah.
We've also stopped the other 99% of species that are actually potentially beneficial to us.
The opportunities to transmit organisms are really reduced.
I mean, just think about the amount of time you spend outside and playing in the dirt, for example.
Yeah.
If you compare...
How much time do you spend playing the dirt?
I spend a reasonable amount, actually.
Yeah. If you keep putting new species in there, if you have all of these ecological niches that are exploited, but then they benefit each other, so you get a synergistic effect of species one is producing something that species two needs. And then you build these networks of interactions.
And I think that's the ecosystem-based analogy or approach that is often under-recognised. We think of single organisms. We think of this bug's that bad, this is good, this is a probiotic with one type of organism. But what you're describing is something far broader and far more diverse that has to be by definition.
And something that's a much more complex
system with interplay
yes.
between everything.
Yeah, that's fundamentally what it is. It's a complex ecosystem, and the environment it's interacting with is us.
Yeah.
And if the ecosystem is wrong, the functions that it should be providing aren't there. We lose the functions, we have the health impacts of that. And it might not be one microbe, it might be a whole community state that produces a particular compound that's then detrimental or absolutely critical to disease.
Yeah, and it sounds like instead of dying of one disease, that has been caused by a pathogen, for example, we're now dying of other diseases because we're losing out on the good bacteria that still have a function. So can you tell us a bit about how we're now experiencing different diseases as a result of the lifestyle changes impacting our microbiome?
That's the key element. Where previously people would have died of infections relatively young, we're talking about them dying in childhood of infections. But the difference now is because people are surviving, but they're not getting these diverse microbes, we have all these diseases that we talk about as diseases of the Western world. And I mean, many of them that, Rob, you treat, the IBDs, the IBSs, the inflammatory bowel disease, irritable bowel syndrome.
Commonly diseases that are described as auto-inflammatory or autoimmune. So essentially, in the absence of the right immune tolerance or the right priming of your immune system with tolerance to the outside environment, there seems to be a dysregulated or an imbalanced immune response whereby you begin to, turn on your own tissues. Or the other opportunity there is for allergy, where you have an exacerbated response to an environmental stimulus because of lack of tolerance to it, because your immune system is improperly developed because of this loss of gut microbiome that we've seen in modern life. And both allergies and autoimmune diseases are skyrocketing. We're seeing just unprecedented rates. So we've moved from life-threatening infection from pathogens to now debilitating and potentially life-threatening autoimmune, auto-inflammatory and allergic illnesses. Yeah.
If we think about it on an immune system scale, we've gone from our immune system not being good enough to our immune system actually just not knowing when it should be good.
Yeah,
so
it's a good way to think of
we've
it.
stopped having it be able to understand, well, this is normal.
Yes.
And then suddenly it gets any signal and it's like, wow, I've never seen this before. This must be bad.
Bull at a gate, I'm going
Yeah.
for it. And then once it's established, just self-perpetuates and you've got something that's a chronic inflammatory condition.
So it sounds like there's an opportunity here through changing our microbiome, we might be able to cure things like autoimmune diseases if we can retrain our microbiome.
If you're putting the bacteria in, you can be reducing the symptoms to a point where it actually doesn't affect your daily life, and therefore it's controlled.
Yeah.
And that's where the opportunity lies. It's something that if we understood it, we then have the opportunity to modify it. And that's the other key thing we have to remember. It's a system that we can modify.
I'm the mum of a 4-year-old and a 7-month-old, and from what I've read, there's pressure to inform the microbiome in a way that will set your child up for life by the age of 3. Is the microbiome fixed by the age of 3, or?
You're born as a blank slate. There's no microbes there, but there's a niche that microbes can colonise. And it almost doesn't matter where those microbes come from. It's the composition that goes in. And we're immediately exposed to a whole range of bacteria. Obviously, typically because of contact, and the mother is a major source of those microbes. So it can be during delivery, it can be later through skin contact or through like feeding. The delivery method matters. So if you're in a hospital environment, if it's a caesarean section, for example, where the mother's had antibiotics, the exposure to those health-associated bacteria are limited, that can open an opportunity for the hospital-associated pathogens to get in as the first colonisers. Through contact with the mother, with the father, with other parents, the opportunity for colonisation is there. Then we go through that early stage where it's predominantly milk. So you see bifidobacteria as probiotics because they're things that we find in breast milk. They are important. And then you start introducing solid foods. And suddenly all the nutrients that are available allow different species to colonise. And so it's all about the environments we're exposed to. And if you think about it, anywhere that you could pick up a bacterial pathogen, you're probably picking up health-associated bacteria as well. So, we think about water sources, we think about food, we think about surfaces, all of these things are potential.
All around us and thereafter within us. Yeah.
And so your immune system is actually developing through those early months of life as well. And so it's this interaction, and this is what we're talking about in terms of exposure to the environment, understanding what is the environment, training the immune system. It's getting the right species in there because that's about training the immune system that these things are okay. You don't need to overreact when they're there.
Right.
So I love it. It's a window of hope.
Yes.
There's so much capacity to take on organisms and so much opportunity to do so that it's not a set situation. I think it's fantastic.
It's not like there's one opportunity for this colonisation to happen and suddenly if you miss that, it's all over. It's just about understanding these things so that the infant gets the best possible chance by being exposed to all these microbes.
Yeah. It's really interesting, Sam. So we're saying there is more instability inherently in those first few years, but thereafter in life, there is also a huge capacity to change your gut microbiome. What is the biggest factor then that's influencing the gut microbiome in someone who's beyond 3 years of age?
Certainly diet has a role. The nutrients that you're putting in there have an effect.
Does it matter where the food comes from? Does it matter if the food's washed, if it's organic, if there's soil on it?
What we know with certainty is processed foods do not have any bacteria.
Processed and packaged, no bacteria, they're sterile by definition.
I mean, if you want your food to last on the shelf for weeks or months,
Not have spoilage.
you don't want spoilage. You want to get rid of as many microbes as possible. What we also know is at the other end of the extreme, fresh foods carry a lot of microbes. And so that's an area that we're actively trying to understand whether the diet-related microbes are actually able to colonise and help us, get that diversity back. because depending on the metabolites that are coming from our host cells, different species will benefit. So, for example, the mucus layer that separates the epithelial cell from the bacteria.
The epithelial cell being the lining of the gut?
Correct,
Yeah.
yeah. And so that the separation there,
Yeah.
that the composition of that mucus will select for some species, will give them the nutrients they need to grow, and others it won't. And even that composition changes over time.
Yeah.
So there's all these dynamic interactions, different locations in the gut, different immune states, different dietary factors. And then with more recent work, we're seeing these interactions with things like exercise, where there's certain factors that are produced during exercise that then modify the immune state, that then modify those exposures for the microbiome.
I love it. It's a bi-directional thing. So as the host, you're exerting an influence on who lives in your gut, and equally the gut
exerting an influence on you.
on us. Sammy, you've been on this journey. I keep going back to the journey, but there's all these technological improvements and you can identify these organisms. We're talking to a man here who's cultured the unculturable. So
Yes,
I think
what do you mean by that?
What I mean is that the breakthrough that Sam and colleagues published was this enormous discovery where we weren't able to culture many, if not most, of the organisms that live in our guts. And Sam was able to successfully do so.
How did you do that?
With metagenomics, like you're looking at the genomes, that's just a big library of fragmented DNA that is only made possible through cultures of actual bugs. So you need to ID the organisms to be able to actually put together these computational libraries of genomes. That's a complex thing to say.
I know, I was going to say, what do you mean by that?
What the hell do I mean? I don't even know what I mean. But what I'm saying is that when you culture bugs, it's the breakthrough that you need to be able to understand who's there.
Yeah, I mean, I think the way I think about it is it's a jigsaw puzzle. But it's a jigsaw puzzle not with one jigsaw puzzle. It's a jigsaw puzzle with 1,000 different jigsaw puzzle pieces mixed together.
That's metagenomics.
That's metagenomics.
Yeah.
What we can do is we can actually say, well, let's grow each of these individual bacteria. And then suddenly we've got, instead of having all these random pieces from multiple jigsaw puzzles, we've got the picture on the front of the jigsaw puzzle box.
Yeah.
So we know the pieces that match this one species.
Yeah.
And we've got that for thousands of different jigsaw puzzles. And so every species we can look at that one piece and we can say that's the right jigsaw that it came from.
Without giving away your secret sauce, key aspects to your culturing, what's changed? We didn't really realise for a long time that these organisms in the bowel don't like oxygen.
We've known for a long time that they're anaerobic, but our ability to actually be anaerobic in the conditions we're growing is really difficult. So some of these species, we actually don't know how long they survive in oxygen, because in the time it takes us to take them out of the environment we grow them in and put it back in, so less than 5 minutes, they die. Wow. So any oxygen exposure they just don't do well with. And so things like that, thinking about the nutrients that they need, and all of this work, it's not just me. Obviously, there's a huge amount of with all science. It's lots of work, lots of history that goes into getting the right place at the right time to bring those elements together. And at the Sanger Institute in the UK, what we actually did was we managed to grow a range of bacteria. And the key thing, by unculturable, it's not that they're never able to be cultured, it's just they haven't been cultured yet.
Yeah.
And I think we think unculturable, it's never going to happen.
Yeah.
Sometimes it's just spending the time to work out this is how you do it. And now we can grow the majority of the species from the human gut.
How important is this understanding to medicine as a whole?
If you're going to make medicines out of these bacteria, you actually need to have the bacteria. That's the fundamental. You can have a genome sequence and say it would be really nice to put this species in, but unless you can purify that species, grow it in the lab, produce it so that you can put it in a pill or put it in a liquid for people to take, the fact that a sequence would be useful is knowledge.
How will culturing these microorganisms and creating this library change medicine?
Yeah, so we'll have these microbes, we'll be able to grow them in the lab, and then we can make mixtures of individual bacteria or multiple bacteria, whole communities, and say, today you've just taken antibiotics. This is a standard community that will give you all the health benefits that we can provide. In a single tablet, you take this, those species colonize, and those species then might be replaced over time. So there might be a different species that's better for you, and you'll be exposed to that in the next week, month, whatever. But you'll have that starting community, you'll have the structures, you'll have the interactions going on, And it's just about giving you that framework to then provide a basis for the colonization with
These are the keystone species you're talking about, those species around which the remainder of the microbial colonies gather and become stable.
So we like to focus on this concept of keystone species, where there's a handful of species that either produce key metabolites that are, or nutrients for other species to grow on, or use something that's going to be detrimental for the community as a whole, and therefore allow the rest of the community to exist. So you need that broader community state. But the key thing is the species that we put in, or the strains of those species we put in, might not be the absolute optimal ones for you. But natural selection will fix that, because if there's a better strain, it will outcompete the one that we've put in. And the other really neat thing here is, even if you've got half a microbiome, the right species that will fill the ecological niche are the ones of, say we've got a list of 100 and you need 50. Those 50 will be available to you, and the rest won't be able to compete, so they'll pass through. So we talk about personalised medicine. And this is sort of personalised medicine. Every individual will need a different selection of bacteria potentially. And so they're getting a personalised treatment that's what they need. But unlike traditional personalised medicine, where it's been designed specifically for them, in this case we've put the whole mix in, and just the things they need are the ones that have colonised. So it's microbial personalised medicine.
And so you're very much moving away from limited strains, small numbers, single organisms, which is what's commonly on the shelf today. Despite the advertising, you're saying that the future is something far more diverse.
Yeah, it's a numbers game. We've just said there's 1,000 species in there. Probably 5 to 10 of them are the ones that are used as probiotics at the moment. The ones that we could grow well in the past, the ones that we could easily purify, may not be the ones that are always beneficial in all circumstances. And I think we have to be careful about the species we're actually talking about here. So bifidobacteria, they're common in infants. They might make good probiotics in infants. But if you're an adult and you're not consuming breast milk, maybe not a great probiotic for you.
You'd have to worry.
It's probably, yeah. And similarly, the lactobacilli, they're common in mouse models, for example. They are found in the small intestine, but not at high levels in adults, certainly. Yet, that's the other group that we're using predominantly as probiotics. That's not to say that they can't have an effect. Even something that's not supposed to be there might stimulate an immune response that then changes the microbiome and is beneficial.
Yeah.
At the moment, we've got all our eggs in those baskets.
And your culturing is unlocking what could be the new therapeutic avenues. You're saying these cultures can actually be used and culture together to make therapies that can improve people's health.
Absolutely. And that can range from just saying, here's a handful of species that are different to the ones we're using and are more beneficial, right through to here's a diverse community, something more akin to fecal transplant.
Yeah.
And anything in between. The problem at the moment is we have to build the knowledge. So, for you, after this antibiotic treatment, this is the right mix of bacteria for you to receive. And that's the direction we're going. And I would say in the next decade, that's where we'll be.
So the next decade in modern life, we're going to all, in some way, shape or form, be undergoing microbial manipulation with organisms to replenish our gut microbes and improve our health.
I think so. I think we're already undergoing microbial manipulation.
Just negatively.
Think about everything. I mean, I guarantee most people have taken antibiotics. Most people have changed their diets. Most people have, moved somewhere in their life.
Yeah.
And all of that is microbial manipulation. It may be beneficial, it may be detrimental. At the moment, we basically put a blindfold on and say this might change things. The difference, though, is we'll do it in a rational way. It will be, this is the intervention you've had, this is the disease you have, this is the risk you have.
Yeah.
These are the microbes that would be beneficial.
Are we re-engineering the gut microbes here? Is that this future world you're describing?
It's a possibility. I think it's optimizing rather than engineering. I don't think we're going to completely say, take these antibiotics, restart. Hopefully you haven't gone that far off track that that's the response you need.
Yeah.
I think it's more about saying, well, we could get an extra benefit here. And, in certain circumstances, for example, like high performance athletes, maybe that extra percentage really matters. I mean, for myself, perhaps less. I don't think I'm going to be a high performance athlete anytime soon.
Yeah.
But you might be able to get a small benefit in terms of health that actually over a long term reduces your risk in a meaningful way. there's all these interactions going on. The metabolites are being produced. They might influence directly immune cells. They might influence other cells in other places in the body. And if the microbes produce a metabolite or any compound that can then go through the body and have an effect, you change the microbe, you change that effect. The problem at the moment is we don't understand most of those interactions. If we understood them, then sure, we could put the right microbe in to say when Rob goes on his run in the morning, he can get the perfect response. He only needs to do 10K instead of the longer run, and you're good.
Yeah.
10K isn't the longer run?
Feels that's pretty long to me.
So it sounds like there's an opportunity here through changing our microbiome. We might be able to affect disease, and things that we thought were fixed might actually be treatable.
That's the opportunity. That's where we have the real ability to revolutionize medicine. We can go from maybe this might have an effect to this is the precise mix of microbes that can give you, whether it's a 10% improvement in a response to a therapy, right through to here's the treatment that's going to mean you never experience that symptom again.
So you're talking about a personalised microbial therapy. That's the world.
It doesn't necessarily need to be personalised.
Because if you go broad enough, you don't have to personalise.
Precisely. But at least this is the right mix of microbes, and it's going to have a meaningful effect on your health.
Well, what a terrific episode. Sam, thank you so much for joining us. It was awesome to have you with us. I learnt so much.
I learned a bit. No, it was
excellent.
It was fantastic. Thank you so much.
Yeah,
for
you
coming
really
in.
just showed us and talked through the complexity of the gut microbiome. It's definitely not a simple thing, but I think the way you've articulated it was just awesome. So thank you so much. It's been awesome having you here.
Thank you.
Thanks, Sam. Have you got the test results back yet?
I do have them back. Should we have a look at them?
Yes, please.
Yeah, let's do it. So what it shows is that your gut microbial diversity is in range, and that's terrific. Gut microbial diversity refers to the variety of species in your gut. The second sort of key metric here, or key point that I'm looking at, is the microbial richness. This looks at the total number of organisms. That was characterised as borderline, interestingly.
Ah, right. So if someone at home has ordered a microbiome test and they don't have a world-renowned gastroenterologist to interpret their results for them, what should they do?
I think it's really important to take those test results to a recognised health practitioner who has expertise and knowledge in the gut microbiome, who could provide you with good recommendations for what to do about those tests.
And what do you think I should do?
I think a dietitian is a good port of call. I think diet day to day has an enormous influence on our gut microbiome, and it would be really terrific for you to get in touch with a recognised dietitian who has an interest in the gut microbiome.
Excellent. I look forward to it.
The advice given in the Gut Truth podcast is general in nature and is not a substitute for professional medical advice. Gut Truth is brought to you by the Hospital Research Foundation and produced by Good Hope Productions.